Good morning everyone. So today seed germination, and also we discuss seed dormancy, and also in this video lecture we discuss how to overcome seed dormancy. So first we will discuss how seed get germinated. Before germination can begin, the seed require three essential external factors. Three essential external factors. They are: first one, water or moisture; second factor or oxygen; and the third one, suitable temperature. So these three factors are essential for seed germination. So how these three factors are essential means, generally here water or moisture, they are required for seed to activate enzymes, and also they soften the seed coat. And how air and oxygen is essential for seed germination means, so due to presence of air, that is oxygen, they perform aerobic respiration to produce energy in the form of ATP, adenosine tri, and this temperature is also essential because it helps to speed up cellular process. There are various biochemical cellular process occurring during seed germination. So to speed up those cellular process, there is a need of suitable temperature process. Now we will discuss each and every step by step how seed get germination. Yes, step by process of seed germination. Generally there are six steps are involved in the process of seed germination. Six steps. Now first we move on. First step here, that is imbibition. Also we can use another term, absorption of water. And here in imbibition and absorption of water, first we need to know what exactly mean of imbibition. Here imbibition is a special type of diffusion. Imbibition is a special type of diffusion process. And here the water is absorbed by, in imbibition process, water is absorbed by the solid. Here the solid structure is nothing but seed. So when the water get absorbed by imbibition process, then this seed cause the enormous increase in value. Increase in value. For example, if the seed will be like this, so when there is a water will be absorbed by the seeds, then the seed will become increase in size or value like this. So this process is called imbibitions. Now we move on second steps. After absorption of water by the seed, then it enter into second steps, that is activation of enzymes and respiration. Here in these steps, so here the absorbed water, here the water which activates metabolic enzymes, very important. So water is required for the seed for activate the metabolic enzymes. Here the metabolic enzymes like amylase enzyme, and also which activate the protease enzyme. When this amylase and protease enzymes get activated in presence of this water, then we break down the stored starch into soluble sugar, and when it break down the stored protein within the seed, then convert them into amino acids. Likewise, there is a need of water to activate these enzymes. When these enzymes get activated, then which involve in breakdown of larger stored molecules into smaller molecules. Like here, stored starch get converted into soluble sugar, and stored protein converted into amino acids to feed these soluble sugar and amino acids. Then later we feed the growing embryo. So this is all the process, all these steps which occur due to presence of this water, because this water get activate these metabolic enzymes like amylase and protease. And here respiration, and after that there is a process called respiration will occur, in which oxygen is essential. Here in which oxygen is required for aerobic respiration. Oxygen is required for aerobic respiration to produce the large amount of energy. In the form of ATP. And now this energy is needed for seed for its cellular division, and which activate the growth during seed germination process. So for that there is a need of oxygen to perform all this process. Now then after that then which move on in third steps. So due to all those process and there is a structure which is arise, so that is radicals. Here third steps, here I am writing emergence of radicals. A radical are nothing but it is a platelet which is developed into root structure. Yeah, see, in which embryo uses the generated energy by respiration process, uses the energy which is generated by the respiration to grow. So this embryo uses this energy to grow. And here it is the first structural part to break through the seed coat, and it is the first structured part to break the seed coat, and that which is emerge as a radical, and these radicals grow down in the soil due to this root shows positive geo, and then this from this radical the first structure which is formed, so that is root system will be formed, root system will be developed. And then after soon after the radical emerge, then there is a emergence of the plumule. We will discuss fourth step, emergence of plumule, that is shoot system. Yes, here soon after the radicals emerge, the plumule begins to grow, and this plumule grows upward towards light, hence it shows positive photo. So hence from this plumule, and then it develop, this plumule develop into shoot system. These shoot system include like stem and leaves and various branches of the plants. So likewise there is emergence of plumule of soon after the radical which emerge. And then we move on. Here we need to know that, so what exactly this plumule and radicals. So to know this plumule and radical, we should know the structure of seed. Here I am explaining the structure of seed. Yeah, here I am explaining structure of seed. Generally the seed is developed from the ovule develops into seed, and ovary develops into fruits, we know that. And here this seed which is made up of with two important components. One is seed coat, another one is embryo, like this. For example, if you consider it is a seed, and around the seed we have, sorry, here, yes, around the seed here like this we have seed coat. See, this is what seed coat, and this rest of this structure is called embryo, and this embryo plus seed coat together called seed. Now here the seed coat, which is a two layer structures, means in which outer layer is there and inner layer is there. The outer layer is called testa, the outer seed coat layer is called testa, the inner seed coat layer is called tegmen. Very important. The inner seed coat layer is called tegmen. The outer seed coat layer is called testa. And now coming to embryo. Generally, embryo consists of two important parts. This embryo consists of two important parts. They are embryonal axis and very important, that is cotyledon. This cotyledon stored food material till the plant prepare, the plant leaves emerge and develop, and to prepare their own food by process of photosynthesis. Till now the cotyledon serve as a food material for the developing embryo. So here the cotyledon and this embryonal axis together called as embryo. Here generally this cotyledons is differ in different plants. For example, in dicotyledon plants there are two cotyledons. In monocotyledon plants there are only one is present. Generally here monocotyledon plants examples like Liliaceae family members and Gramineae family members comes under monocotyledon. They contain only single cotyledon. And coming to dicotyledon plant, they contain two cotyledons. For example, here Fabaceae and Solanaceae. So this Fabaceae and Solanaceae, these two comes under dicotyledon plants. They contain only two cotyledons. But Liliaceae and Gramineae, they comes under monocotyledon. They contain only single cotyledon. Now here coming to embryonal axis, and this embryonal axis generally which contain two end, here upper end and lower end, and here the lower end contain radicals. Very important. The lower end contain radical structure, the upper end contain plumule structure. Now here with the help of diagram I will explain how this is present means. Yeah, here for example here I am explaining. See, this is the embryonal axis. Think it is the embryonal axis. From this embryonal axis there are two cotyledons like this. These two are cotyledons, embryonal axis. The lower end of the embryonal axis, here we have one structure and here we have one more structure. And now this structure is called radicle. And here the upper end of the embryonal axis called plumule. And these two are what? Cotyledons. And plumule and radicle is connected with the help of embryonal axis. Means embryonal axis plus cotyledons together called embryo. And this embryo later which is covered with a protective sheath, that is seed coat, like this seed coat. Now it become a complete seed. See, this is what? Seed coat. And embryo, the rest of the structure inside the seed coat is called embryo, and that embryo contain two part. One is embryonal axis and two is cotyledons. And this embryonal axis lower end contain radical structures, upper end contain plumule structure. And then this plumule later develop into shoot system, and this radical later develop into root system. So like that, first in seed germination, the radical which is emerge and later it develop into root system. Soon after, the radicals which develop, then plumule gets started emerge, and which is later develop into shoot system. So this is what exactly mean of plumule and radicals. So after formation of both root system and shoot system, then it enter into fifth stage, that is development of, then that structure is called seedling. Now here fifth one is development of seedling. Here, sorry, yeah. So depending on the type of seed, here depending upon the type of seed, the germination follows one of the two pathway, any one of the two path, either hypogeal or epigeal germination. For example, here we need to discuss two pathway for development of seedling. One is epigeal germination and second one is hypogeal germination. So this development of seedling follow any one of the pathway. It is completely depending upon the type of seed. So here in epigeal germination, what happen means, here in epigeal germination, the cotyledons are pushed above the soil like this. For example, if it is a soil, so here we have seed get seed, and now here seed start its germination like this, and then there is a stalk like structure, that is embryonal axis structures, and this embryonal axis structure which push the cotyledons above the soil. So here the cotyledon will be appear here like this. Here see this structure, the development of like this structure is called epigeal germination of seedling. So here I am writing, so the cotyledon are in which these two are coty. So here the cotyledons are pushed to the soil. See, this is what soil, and here we have seed above the soil. This type of germination of seedling generally occur in various plants like castor and bean plant, and very important tamarind. And now coming to hypogeal germination, in which the cotyledon remain below the soil. Here below the soil, in which cotyledon remain below the soil. Here means in below the soil only the cotyledons are remain. And this type of hypogeal germinations are generally seen in like pea plant and what, yeah, maize, pea and maize plant. So likewise, any depending on the type of seed, the development of seedling occur any one of the pathway, either epigeal germination or hypogeal germination. In epigeal, the cotyledons are above the soil. In hypogeal, the cotyledons are below the soil. So this is how the seedling get germinated by this process, by this pathway. Now we move on. The last most important, so that is formation. After development of seedling, then it undergoes formation of green leaves. Yeah, sixth step, formation of green leaves. Here, as shoot reach the above the ground, as shoot reach the above the ground. Generally, shoot system develop above the ground and root system develop below the ground or soil. As shoot reach the above the ground or soil, it develop into first true green leaves. It develops into first true green leaves. So then it begin photosynthesis. Then these green leaves start producing its own food by photosynthesis. When there is a formation of green leaves, and then it start producing its own food by the process of photosynthesis. Till the formation of green leaves, the food material for embryo development is served by the cotyledons. So this is all about the how seed get germinated. And now then we move on very important concept, and sometime the seed will undergoes dormancy, means they won't get germinated. So that is the concept of seed dormancy. Yeah, here I am writing about seed dormancy. So here what is seed dormancy? Yeah, seed dormancy means inability of seed to germinate even after appropriate condition. Even after providing appropriate condition, if the seed doesn't germinate, such condition is called seed dormancy. So inability of seed to germinate even after providing appropriate condition, then the seed said to be as a dormancy. What are the causes for seed dormancy? Here there are few causes for seed dormancy we have. Now we will discuss. So here the first and most important causes of seed dormancy here, that is seed coat abnormalities. Seed coat become abnormality. It means that if the seed coat be tough and hard and tough seed coat will be there, in that situation the seed not undergoes germination, or if even though impermeable seed coat for the water and oxygen, to water and oxygen or air. And one more important causes for seed dormancy, that is there are some inhibitors like chemicals, they inhibit the seed to become germinate. So inhibitors are the chemicals, like there are few inhibitors are there, like abscisic acid. It is one of the most important inhibitor of seed germination, that is abscisic acid. It is a phytohormone. And one more, that is ascorbic acid. There are also phenolic acid and ferulic acid. So these are the few different chemicals, they inhibit the seed to become germinate, means they are promote seed dormancy. All these abscisic acid, ascorbic acid, phenolic acid and ferulic acid, which promote the seed dormancy, it means that which inhibit the seed to get germinate. And one more important causes for seed dormancy, here I am writing third one, that is immature embryo. Means here embryo not properly developed. In that condition also seed not germinated. Improper development of, or embryo not properly developed. Which embryo not properly developed. So these are the three important reasons for seed dormancy. Now the most important thing is there is a solution for problem. Every problem has a solution. Likewise, and these are the various problems for seed to become germinate, and there is the solution is there, and we can overcome this seed dormancy. How means we will discuss. Yeah, yes. Here, overcome or breaking of seed dormancy. Overcome or breaking of seed dormancy. Here, how to overcome, how to break means here we will discuss. By four important methods here we use to break or to overcome this seed dormancy. In the first and most important method, that is scarification, means in which weakening of seed coat. Here we need to weak the seed coat, weakening of seed coat. And this weakening of seed coat done by two methods, either mechanical, then it is called mechanical scarification, or we can done by chemical, by using chemical, hence they are called chemical scarification. Here chemical we use to weak the seed coat, like sodium hydroxide and sulfuric acid H2. And here there are some chemical scarification in which we use sandpaper. Means by using sandpaper we need to rub the seed against the sandpaper, then we can weak or we can lose the seed coat. So likewise we can use these two, mechanical scarification and chemical scarification, to remove or to weak the seed coat. So that complete process comes under scarification. Now second and most important method, or method to overcome or break the seed dormancy, that is stratification. Very important, that is stratify. So this stratification also called pre-chilling treatment. So in this method here we use low temperature. Here we use, here I am right, just wait. Yeah, here in pre-chilling treatment or stratification, we use low temperature treatment for seed germination. And here there is a one important note. Sometimes they will ask in exam and they will confuse. Here there is a low temperature treatment for flowering. Note, low temperature treatment for flowering is called Varna. So here we can use low temperature for two purposes. One for seed germination, another for flowering. The flowering concept is called vernalization. Now coming to third and most important method to use or to break the or to overcome the seed dormancy, that is impactions. Third one, impaction. The impaction means in which what we need to do means vigorous shaking of seed to unblock micropyle. Vigorous, in which we need to do vigorous shaking of seed. Why means to break micropyle means there is a one important pore-like structure or pore-like structure in seed. So we need to break those pore. Then we have to do vigorous shaking. Such concept is called impactions. Now last and most important method to overcome or break the seed dormancy, that is chemical treatment. So yes, so fourth and last important, chemical treatments for overcome the to overcome the seed dormancy. So here we use few chemicals like gibberellic acid, very important. Gibberellic acid. Generally, gibberellic acid is a phytohormone, and this phytohormone is also known as seed germination hormone. Seed is a seed germination hormone. If we can treat the seed whose become dormancy with the help of gibberellic acid, then seed undergoes germination. So, very important chemical treatment for seed germination, that is gibberellic acids. Now coming to second one, that is thiourea. We can also use thio for seed germination. It means that it breaks the seed dormancy. And third, that is potassium nitrate. We can also use potassium nitrate to overcome this seed dormancy and to become the seed germination. So all these are various chemical treatment we use to become seed germination and which inhibit the seed dormancy. And there is a one important note here, and sometimes they may be asked, that is note here, and inability of seed to germinate due to external environment condition, and such seed dormancy is called quiescence. For example, here the external environmental conditions like improper providing of light, improper light, a temperature, and insufficient of water and oxygen. So these are the external factor: light, temperature, water, and oxygen. If there is a improper means, then seed also undergoes dormancy in condition, and such concept is called quiescence. If inability of seed to germinate due to in factor, then it become dormancy. If any external factor which influence the seed to become dormancy means, the concept is called quiescence. So this is the major differences between dormancy and quiescence. In dormancy, internal factor only; in quiescence, the external factor only which leads the seed to become dormancy. So this is all about the entire concept of seed germination and seed dormancy and how to overcome this seed dormancy. So with this we complete. That's it. And here I have provided three highly important NCERT based MCQs from here. So yeah, consider the following statement regarding the seed germination. So here asking about seed germination. The first step in seed germination, read all these three statements. Which of the statement given are correct? So answer it and drop your answer in comment box. Don't forget. And one more MCQs here. Yeah, read the following statement about seed dormancy. And first MCQ asked about seed germination, and this MCQs asking about seed dormancy. So read these three statements and which of the statements above are correct. One more MCQs. Yes. Yeah. Here, evaluate this statement considering the method used for breaking seed dormancy. And these MCQs completely frame only how to overcome or breaking of seed dormancy. So read this three statement asking about scarification, stratification method. Already we discussed just now. And read this three statement and answer which of the statement given above are correct. So these are the three highly important MCQs. So with this we have completed. So that's it for today. So if you like my videos, please hit the like icon button and share this video to your friends, and don't forget to subscribe my Mahadyuth Academy channel. So thank you and have a nice day.